research that my lab at the Scripps Research Institute is engaged in. And it's always the most distinct pleasure when you're invited by the students. And this has been an incredibly impressive meeting, and I'm really impressed with the students that put this on. So thank you, and it's really great. So what my lab works on, I'm trained classically as a chemist, and so somewhere in the middle of grad school, I decided that all the interesting questions were in biology, so I started making that transition. What my lab works on now is directed evolution of DNA polymerases to have novel activities, some evolution-inspired approaches to antibiotic discovery, biophysical characterization of the adaptive mutations that confer function. And what I'm going to tell you about today is our effort to develop unnatural base pairs. It's always weird when I get introduced now and or I go to meetings about genetic code expansion, because I spent so much time where I couldn't use the word code, I had to use the word alphabet. But about 2/3 of the way through this talk, I'll actually be able to use the word code today, so that's exciting. So if one's interested in developing an unnatural base pair, so first, just for comparison, here's natural DNA. This is part of the genome of E. coli. If you can't see it, let me blow it up. I actually gave this talk in China last year, and someone came up to me afterwards and said, you know, you have a lot of nerve showing the Japanese flag in China. So, you know, I think to a chemist, what's remarkable about DNA is how unremarkable as a polymer it is. It's just a simple linear coupling of 4 different covalently linked letters, nucleotides, G, C, A, and T. But of course, what makes DNA so remarkable and so able to uniquely underlie all the information storage and retrieval that enables life is the selective pairing, the molecular recognition, the pairing of G with C and A with T. And so if one's interested in developing a 5th and a 6th letter, 5th and 6th nucleotide to form an unnatural base pair, to increase that, the potential information that organisms could store and eventually retrieve, there's a lot of things you have to be curious about, you have to be worried about. And so here, this is my rendition of a bacterial cell. I'm a chemist, but you can see my 2 little membranes there, so it's obviously gram-negative. So one would have to be concerned with, first of all, you'd have to have requisite unnatural triphosphates, let's just call them X and Y. You'd have to have them available within the cell. You'd have to have the unnatural nucleotides present in some sort of a genetic element, a plasmid, a chromosome, something. And you have to have DNA polymerases efficiently, when they come across them, efficiently incorporate the unnatural cognate triphosphate, and only that, and then efficiently continue synthesis to replicate the element. Same story with the RNA. You have to have RNA polymerase. Well, you have to have the ribotriphosphates available for X and Y. You have to have RNA polymerases efficiently recognize them to produce both message and tRNA, which then have to assemble at the ribosome to mediate decoding of those unnatural codons to produce proteins with, purple balls in them. At this point, I always wanted to note Peter Schultz's work. Because right from the beginning, I was a postdoc with Pete, and right from the beginning of this project, I planned on stealing Pete's work. So what Pete did, for those of you who don't follow this field, was that he expanded the genetic alphabet by taking amber stop codons, which are naturally suppressed in nature under various conditions, and finding an orthogonal tRNA synthetase pair from a different organism, an archaeal species, and where that synthetase could be modified to charge its cognate tRNA with an unnatural amino acid. And then if you use that in E. coli, you could get these tRNAs charged with unnatural amino acids, and then he recoded those tRNAs to suppress amber stop codons. So that way he could encode a 21st amino acid. And it's absolutely beautiful. And my personal opinion is that I think it's a pity, it's a joke that he hasn't won the Nobel Prize for that. I think it's absolutely beautiful. I think it's the first, and to this day, best, what we would call synthetic biology. But it's not perfect. I can say that because Pete's not here. It's limited because you only got one unnatural. And most problematically, it's an amber stop codon. It's still competing as a stop signal. It's still recognizing release factor, so you get truncated protein. People are trying to eliminate that and recode the genome, but then you have to use rare codons, and it turns out that when you eliminate release factor, you're eliminating competition for suppression with that release factor, but you're also increasing competition with all near cognates. So it turns out fidelity drops significantly. So the idea was simply, look, why reappropriate what nature created? If you want something new, then just go to the ground floor and build it. So that was the idea, to create an unnatural base pair, which would allow us to create more unnatural codons than we could ever use. So that was the idea. So the approach that we took, well, okay, first, excuse the drama here. I want to give a couple, because I actually get asked this sometimes, and to me as a chemist, it seems so axiomatic that this would be cool to do. But I get asked, why would you want to do this? And so I'd give two answers to this. One sort of philosophical or conceptual or whatever, and then a practical motivation as well. So conceptually, I think it's fair to say that people have thought about life and the molecules that underlie life since people have had thoughts. And I think that certainly a lot of people have interpreted that as evidence for a creator. Things, what the chances are that we could evolve like this? We're so perfect, and so there must be evidence of a God. And I think that a lot of people believed that for a long time. In more recent times, I think, from a more scientific perspective, people have thought about more mechanistic scientific approaches to that problem, answers to that problem. But whichever approach you take, sort of an evolution scientific approach or a religious approach, I think it's fair to say that you'd think it's pretty unlikely that chemists could come in and build new parts that function within and alongside the natural parts. Evolution's been tinkering and modifying and optimizing them for, 4 billion years? God knew what he was doing when he designed them. But how perfectly designed or optimized are the natural molecules of life? We have nothing to compare them to. So I think that's a really interesting question. A more practical perspective, protein drugs have been, they've been around for actually a long time. Insulin was introduced, 40 years ago, actually from cows, like 150 years ago or something. But recombinant insulin was introduced and has revolutionized medicine. There's been another revolution in protein therapeutics with antibodies. And I think that if you compare protein therapeutics to small molecule therapeutics, I think there's a really interesting story there. Modern medicine basically got started when medicinal chemists figured out how to purify the critical active molecules from the plants and the things that people had empirically determined had medicinal value. And they figured out how to purify them, they figured out how to administrate them. But modern medicine really got started when synthetic chemists learned how to modify those molecules to make semi-synthetic variants, because those natural products didn't evolve to be drugs. They didn't evolve for PK. They didn't evolve for stability in human serum. So, this led to all the classes of the antibiotics that we have today. For example, the beta-lactams are all now semi-synthetic variants of the original beta-lactams that were discovered. And on top of that, if you look at the sort of molecules that medicinal chemists have built for other therapeutic indications, they're incredibly diverse. They're much smaller than proteins, but they're much more diverse than proteins. And those diverse functionalities within those small molecules are what make them great drugs to treat different diseases. And the access that chemists have to different functionality is much greater than that provided by the amino acids that you can build a protein from. So if one could expand the repertoire of monomers that you could use to build proteins, maybe even choose amino acids for a desired function. One of the most common pharmacophores in small molecule drugs is electrophiles. How many electrophiles do amino acids bring to the table? 0. So it would be really cool, it would be really interesting if you could start developing protein drugs that have electrophiles and all sorts of whatever sort of functionality you're interested in. So that's a conceptual and practical motivation. So the way we went about it, I forgot to set my timer. Passed. The way we went about it was that we thought, well, look, nature uses complementary hydrogen bonding to underlie its pairing of G with C and A with T. We, from the very beginning, worried that if we tried to develop an unnatural base pair based on hydrogen bonding, that they would mispair with the naturals, which we thought from the beginning would be our biggest challenge. And so what we wanted to do was we wanted to ask the question of whether a different force could be harnessed to underlie that information storage. And so everyone, I think, is familiar with the hydrophobic effect. Oily things like to get out of water. They like to aggregate with themselves. In addition to that, when they do aggregate with themselves, they interact with each other through London dispersion forces, induced dipole, packing forces, whatever you want to call them. And those are strong, so we wanted to ask if we could harness those sorts of interactions to mediate selective and efficient pairing of two unnatural nucleotides in the context where we have some basal selectivity, orthogonality against mispairing with the more water-like hydrophilic natural nucleotides. So all of the analogs that we've explored are hydrophobic, predominantly hydrophobic. So that's one thing I want to say about them. In the early days, they were also large because we wanted to stabilize things, because if you're taking the H-bonds out, you got to do something. So a lot of them were large. We pared them down in later generations, as I'll show you in a second. So one thing I want to say about them is they're all large and hydrophobic. And of course, I'm not showing the sugar or the phosphate. These are just the nucleobase part. A lot of them aren't even basic, but I'll just call them nucleobases for simplicity. And so these are just like the GCAT components of the nucleotide. They're all synthesized as triphosphates and as phosphoramidites. And so one thing I want to say about them is they're all large and hydrophobic for reasons that I just described. The other thing I want to say about them is there's a lot of them. We did not want to take the sort of, I don't know what you call it, traditional chemistry approach where we come into the problem and say we very carefully design an unnatural base pair to fit together just right, because we didn't think that we understood enough about how DNA polymerases replicated DNA to be able to do that. So we took a medicinal chemistry approach, and that was we simply started making analogs like this. Literally benzene rings were some of our first analogs. And then we just made modifications and analyzed them. We looked at their stability, we looked at the ability of DNA polymerases to incorporate an X against a Y, the fidelity of that step, and then the ability of DNA polymerases to then continue synthesis after incorporating the natural base pair. And so we literally used that data to develop what medicinal chemists would call structure-activity relationships, and we used that SAR to feed back into the design strategy and make other analogs. That's where these all came from. This is, in the lab, often referred to sort of as the first generation analogs. We've made over 200 to this point. Here's analogs that were designed to be a little bit smaller. You can see that we're systematically exploring substituents like fluorine, methyl groups, halogens, O-methyls. And after an embarrassingly long time, about 13 years, we finally came up with a pair. This was the first generation analog, this 5-methyl sulfur isocarbyl styrol. That's what a chemist would call that. And this methyl methoxy, the second of a methyl methoxy series. This was the first generation analog, that was the second. This came out of a screen. A graduate student and a postdoc screened, independently set up 2 screens for 1,800 candidates. We had 60 by 60. And each of them independently identified the same top hit. It was this pair. That very quickly reinvigorated our design efforts. And we found out that filling out this quadrant right here of MMO2 to produce NAM produced a better partner for 5,6 than MMO2 was. This pair was our first that PCR'd efficiently. So we were really excited. It was sort of a milestone for us because we could actually make DNA templates, put it into a PCR reaction. All the SIR that I described up to now, well, in a second, I'll describe the way we analyze these now, but and so what I'm going to talk about in the next 20 minutes is mostly this pair. The next 15 minutes is mostly this pair. We're continuing to develop, to optimize things, and since we discovered this pair, we've now discovered this thiophene derivative that we call TPT3 as a better partner for NAM than is 5,6. We had this earlier, and so a lot of the results I'm going to tell you about are going to be with this pair, but I'll return to this pair in a little. This pair, obviously this pair and this pair as well, are too efficiently synthesized by DNA polymerases to use steady-state kinetics to measure their rates to drive that SAR that I just described. So we had to come up with a different assay, and I mentioned that they could PCR. So we developed a PCR assay. This was, this looks like data to you. To me, this looks like my second kid, because this was really exciting to see. What we did is we PCR amplified a piece of DNA, and we just gave it to the Scripps sequencing facility, and they ran a standard Sanger sequencing reaction, and this is what they produced. This is the way we analyze all our things now. You see a normal sequencing chromatogram up to the point where the unnatural base pair is. You can read it, you can sequence it, it's normal, and then you see an abrupt termination. That was the first time I felt like I actually saw the unnatural base pair. So that was really exciting. So you can ratio the amplitudes before and after to get a measure of the fidelity during PCR. This is a minimum fidelity because this sequencing reaction doesn't have the triphosphates of our unnaturals present. So that's why it terminates. But it's certainly going to get some read-through during the sequencing reaction itself. That's probably where the majority of this comes from, but the errors during PCR plus the error during sequencing results in this amplitude. So we interpreted simply the error in PCR, and we use that as a minimum measure of fidelity. You can then use calibration curves to determine what the fidelity is. And what we typically find is fidelities range in the 99.9-ish%. These are just the local region around the unnatural base pair. In this case, beta is NAM. In their 180 more fragments. We've looked at many sequences, GC-rich, AT-rich. And the efficiencies are shown here. These are measured by qPCR. We've actually put the unnatural base pair next to each other, two next to each other. It's a little bit harder to get a gauge on the fidelity here because in this case what you're reading is a drop on a drop. So that's a little bit harder to read, but it's still very high. And these are not 100-fold amplifications. These are more than you'd ever have to do. So it gives plenty of chances for other sequences, loss sequences to sneak in and outcompete, and that doesn't really happen. So we were pretty happy with this. We're chemists. 99.99 seemed really great to us, but maybe there's some biologists in the room that are thinking, well, wait a minute, 99.9. I thought DNA polymerases had fidelity of like errors of like 1 in 10 to the 7 or something, 10 to the 8 or something. Isn't that 99.99999? If that bothers you, hold that thought, and I'll come back to it. But we were chemists, so we were pretty excited at that time. So maybe this was our chance to sort of charge into the long-term goal of deploying the system in a living organism. But taking another medchem analogy, we didn't. And the reason we didn't is we got a structure with my friend Tammy Dwyer at the University of San Diego. And here's an average of 10 NMR structures. Here's their average. Here's 10 NMR structures. Here's their average. And if you look, you see our unnatural base pair. It's cross-strand intercalated. We actually knew that they were prone to do this from early structural studies that I haven't had time to describe. But those are with bigger analogs, and we thought, well, that intercalation comes from them being large. We had kind of thought since we'd optimized them, they'd come in like a Watson-Crick pair, because everyone knows that's what DNA polymerase is like. So if you have a pair that's being replicated well, it must mimic a Watson-Crick pair. And we were kind of surprised. And so the reason we were worried about this wasn't structural. I don't care what the unnatural base pair looks like. All I care about is its function. But what worried us, so I mean, the analogy I use, again, is a medchem analogy. If you've got a molecule that, like, let's say, kills Staph aureus, it kills MRSA, and you go to your friend from Pfizer or Merck or Novartis or whatever, you say, look, let's pull down a couple $1 million to start a medchem effort. He'll say, well, what's the target? And you'll say, I don't know. I don't know the mechanism of action, but I can show you it works. You'll get zero interest because pharma has been faked too many times by activities that disappear. So before they pull down the investment to initiate sort of that kind of effort, they're going to want to know that you have a well-defined you know what the target is, and you have a well-defined mechanism of action. So the problem here, the analogy is, look, we know that Watson-Crick base pairs are required for replication. I claim they're replicating well, but they shouldn't be. Something weird going on here. Are we chasing a ghost? So I started a collaboration with my friend Andy Marks at the University of Constance, whose name should be there. I don't know why it's not. Well, let me tell you the problem. Let me reiterate the problem. If you look at a GC, a CG, a TA, or an AT base pair, they all kind of look the same. They look like this Watson-Crick planar structure. Doesn't matter whether you look at the structure in a duplex or whether you look at the structure formed in the polymerase active site by pairing a triphosphate against a template. They all look the same. They look like a Watson-Crick base pair. Ours doesn't look that way. Ours looks like an AA zipper motif mispair, if you're familiar with that. Everyone knows that polymerases evolved to not recognize them. So what's going on? So the way a DNA polymerase works, the mechanism, for those of you who don't think about this every day, a DNA polymerase looks like a right hand. A primer template lies down like this, and only when the correct triphosphate binds, it induces a large conformational change of the fingers domain down over the palm and thumb domains to form a tight closed complex that's supposed to rigorously select for a correct Watson-Crick base pair. And so we had two questions. Number one was the formation of our unnatural base pair sufficient to drive that same large conformational change? And number two, if it was, what is that tight closed complex recognizing? Because our pair doesn't look anything like a Watson-Crick pair. It looks like a mispair that the polymerase is supposed to reject. So we collaborated with my friend Andy Marks, whose name is there, and his graduate student Karen Betts, and we got a bunch of structures. I'm only going to walk you through the couple that are most relevant to the story I'm telling. Here's the binary complex of the primer template bound to the polymerase. The only part of the polymerase I'm showing you is the O and the O1 helix, which form the base of that fingers domain that I just mentioned. When you add and so here's our little NAM on guy, and the template flipped out of the developing duplex. When you add the correct cognate triphosphate, in this case 5,6 triphosphate, he flips back into the developing duplex, and you get this large conformational change of the fingers domain. To illustrate that conformational change, I've overlaid the binary and ternary structures here. Here you can see that conformational change. To convince you it's exactly the same as the conformational change induced by the formation of a natural base pair, here's an overlay of the ternary structure synthesizing the unnatural base pair and synthesizing a GC base pair. You see at the secondary structure level, they are absolutely superimposable. Even at the side chain and the bound waters and magnesium level, they're virtually superimposable. So the answer to the first question is very much induces the very same conformational change in the polymerase. So the second question, and if your eyes are good, you can maybe already see this. And if your eyes are like mine, let me help you. This was a good day in the lab. The unnatural base pair is not replicated with some weird mechanism. A natural base pair is replicated with an induced fit mechanism. Its formation drives a large conformational change of the polymerase. The unnatural base pair is replicated with a similar but only subtly different mechanism. It's a mutual induced fit mechanism. Its formation drives a large conformational change of the polymerase, but that conformational change in the polymerase drives a reciprocal conformational change in the base pair. The base pair does adopt a Watson-Crick structure, but only during replication when it has to, as directed by the polymerase. So we were really encouraged by that. We thought we had something that replicated really well. We understood the mechanism for replication. It wasn't tricky. We weren't getting faked out by anything. So at that point, we were encouraged enough to try to get this working in a living organism. The living organism, of course, of choice was E. coli, due to the tools that are available as the first effort to develop a semi-synthetic organism. We call them SSOs, semi-synthetic because they have a man-made component in their guts that they're using. That's the synthetic component. So the first challenge that you're immediately faced with, and I sort of brushed over this, is how do you get these requisite triphosphates within the cell? And I know anyone who thinks about these problems, I'll take the nucleosides and let them diffuse in. They'll magically be recognized by the kinases of the salvage pathway and magically taken up. E. coli doesn't even have a complete salvage pathway. Many bacteria don't. We tried that very hard. We tried to bring in exogenous kinases from other organisms. The bugs just get sick, almost certainly due to imbalancing the natural triphosphate pool. So the way we solved this, I think, is a great example of interdisciplinary science. And what I mean by interdisciplinary science is you read other people's literature. What I read and got really excited about was that there was a small group of people that studied the problem I'm about to describe to you. That there are genetic elements that replicate autonomously. For example, the chromosomes of various plastids and eukaryotic cells, the chromosomes of a variety of obligate intracellular bacteria. They replicate autonomously. They live in a cell, but they replicate autonomously of the cell's genome. What was really interesting was that several of them, of their chromosomes, or of the genomes, don't encode the machinery of triphosphate synthesis. So where do they get their triphosphates? I mean, they're separated by a membrane from any source of triphosphates, and they don't make their own. Turns out what they do is they produce a dedicated nucleoside triphosphate transporter. So we got really excited about that. We wrote to various labs and got 18 of them and looked at them in our bacterial strain. And we're really excited to find that one worked. And so the idea was from an algal species, PTNTT2. It's the second of a dedicated nucleoside triphosphate transporter that it uses for chloroplast triphosphate uptake. So the idea is that we can now provide the triphosphates to the media. They'll diffuse in through the porins. We actually know it's which porin is the dominant one. Not surprisingly, it's the big one. And then expression of the transporter will facilitate uptake into the cytoplasm where they then become available for replication in a genetic element. So the first, to try the first experiment, we produced a couple of plasmids. One we call PAX for the accessory plasmid, and that's where we're going to put all the accessory elements of an expanded genetic code and eventually an expanded genetic alphabet. Right now, all it has is that transporter that I just described. Then this PIMP plasmid. This PIMP plasmid is just PUC19. It's identical to PUC19, except it's had one base pair, an AT base pair, replaced with an XY. So when we made that PIMP plasmid at our bench, we made it with this base pair. Remember this TPT3? It replicates really well. By this point in time, we knew it replicated well. We just hadn't validated as much as we had. We didn't have a structure on it. And so the plasmid that we constructed at our bench, we constructed it with this pair. But the pair that we used for the in vivo replication was this pair. So the triphosphates of these two were provided. And during the first round of replication, this would simply be inserted against NAM, and you convert that to this pair for your in vivo experiment. So that was the idea. So here's the experiment. This is the first data that we actually collected. I remember this day, I'll remember for the rest of my life. It was a Friday night when my student ran this experiment. Isolated the PIMF plasmid, so transformed it in, isolated the plasmid, PCR amplified it with triphosphates where one of our unnaturals was biotinylated. The reason we did that is it allows us to separate the amplicons into those that came from plasmids that had lost the unnatural base pair and those that had retained the unnatural base pair. And then we did a strep-Evans supershift assay. So if you don't have the unnatural base pair in the template, you transform the bacteria with just PUC19. There's no unnatural base pair in the template. It doesn't matter whether you add the triphosphates or not, you don't get a shift. If you have the unnatural base pair in the template, but you don't add the triphosphates, you don't get a shift. If you have the unnatural base pair in the template and you provide their triphosphates, but you don't provide for their uptake by induction with IPTG of the transporter, you don't get a shift. Only if you have the unnatural base pair in the template, you provide the triphosphates, you provide for their uptake, you get a shift. And the shift corresponds to a fidelity of about 99.9 per division. So we were pretty excited. We wanted an alternative, an alternate way. This was after 15 hours, 22 doublings, and that corresponded to a 10 to the 7-fold amplification in the plasmid. We wanted an independent way to demonstrate that, so here's that sequencing assay that I already described. All the controls are here, and here again, only in the correct experiment where you have everything added, you see this abrupt termination, and you can get a fidelity that roughly matches what I just described. So we were pretty excited. We submitted a paper. On December 21st, I got the response from the journal, and 2 of the 3 reviewers said, yeah, you're in public. The third said, you absolutely have to do an experiment. Turns out that there were 2 things he didn't like. He didn't like this, and he didn't like this. This third reviewer hated PCR. He thought we were somehow introducing the unnatural base pair during the analysis or something. You know, if you want to talk, if you can come up with a way that we could have done that, if you can come up with a way that this could be an artifact, find me after my talk and explain it to me, because I can't. But, you know, reviewers, what are you going to do? So on the 23rd of December, I emailed NEB, New England Biolabs, because I'd seen they had published a paper, Yvonne Corina had published a paper where they used a beautiful LCM-SMS assay to identify a single epigenetic modification in a plasmid. And I thought, well, if they can do that, they can detect our unnatural base pair. He emailed back. I didn't check my email till the 26th. I was busy for a couple days there. And I got this email back, and he said, okay, we've already ordered the triphosphates. They're commercially available. And here's a workflow. Will be done in 2 weeks. They weren't. They were done in about 4 weeks. I can't express my gratitude to Yvonne and his NEB team enough. Because anyone who's tried to run a collaboration knows how challenging it can be. But it took 4 weeks instead of 2 because there were a few little glitches that were specific to the unnatural base pair they had to work out. But here's their data. This is the mass spec data. Here's the C content. Here's the G and the A. T due to ion suppression doesn't fly. This was expected. Right out here, right where it should be, is the exact mass of 5,6. Not TPT3. The only place 5,6 was provided was as a triphosphate to the media. So, since the last common ancestor of all life on Earth, it's been 4 letters, 2 pairs. These cells are growing and dividing. Do I have 5 minutes left?
Yes.
Wow. With 6 letters and 3 pairs. Okay, this 99.9% issue, I'm going to go through this quickly. I'm a synthetic biology lab, I guess, and I think that means I have to use Cas9. So the idea was that we've already talked about Cas9 in a beautiful talk yesterday. We wanted to use it in a slightly different way. We asked if we could design guide RNAs that target loss sequences. In other words, would the unnatural base pair protect the DNA from cleavage by Cas9? And of course, unlike in eukaryotes and bacteria, if Cas9 is active, REC-BCD loads and destroys the DNA element, so it doesn't lead to recombination. It leads to loss of the entire genetic element. So 99.9% of the time when you replicate, it replicates well. That 1 out of 1,000 times that you introduce an unnatural base pair, could we eliminate them with this error correction mechanism? We constructed a plasmid that had 2 unnaturals in an mRNA and a tRNA. You can guess where we're going with that. Ignore the synthetase. We had to incorporate Cas9 into the genome of our bacteria. Obviously the transporters there, and we encoded in the PIMF plasmid now the 2 mRNA guides to keep the mRNA clean. I'm not going to go through this much, but I think you can see this growth regimen. These are each one. These are each 30,000-fold dilutions. This is growth in liquid culture. It doesn't include the growths on 2 separate platings, so there's a massive amount of growth here. You can see the retention here. This initial loss right here is from PCR errors during construction of the plasmid that cloning allows us to clean up. The unnatural base pair replicates with higher fidelity in the organism than in a PCR reaction. We can virtually replicate in any sequence context for as long as you like now, because Cas9 cleaves it. We put the unnatural base pair into a message. Obviously, we wanted to look at transcription. This time we lyse the bacteria after induction with T7 RNA polymerase. We then take the isolated RNA, we reverse transcribe it with notoriously error-prone reverse transcriptase, PCR amplify it with the same byte and tag. Trick, there's the shift. The fidelity is very good. It's obviously transcribing very well. We've now done that in a lot of different sequence contexts, including mRNA, tRNA. The translation piece, I'm going to go through this literally in 2 minutes. The phenotype that we're looking for was in vitro lysates, where we directed the incorporation with the unnatural base pair of an unnatural amino acid that has a click handle on it. We then take that protein and click onto it a fluorophore so that we can identify unnatural content, proteins that have the unnatural amino acid by virtue of their glowing, having the fluorophore. So the assays that we ran through a whole variety of different anticodon-codon combinations to try to pick a few to go with was this assay where if these are in vitro assays, so we're just adding them to a test tube now, but if you produce mRNA with the unnatural codon and tRNA with the unnatural codon, you want a signal because your natural amino acids there, you're clicking on a fluorophore. But if we have the mRNA, we also ran as a control, the tRNA with the unnatural anticodon with the mRNA with the natural codon, because we wanted to make sure that the tRNA wasn't, that the unnatural anticodon wasn't problematically decoding other natural sequences. So that we wanted to make sure it was specific for the unnatural anticodon. I usually have a joke here where I show you all the codons that work, and I say that we picked this one because it was in the upper left. Trust me, it's a really funny joke. But here's the data. Now, this is in vivo, right? This is in protein isolated out of cells. This is the codon AXC, X is NAM. We're adding the amino acid azitolysine, because that's the click handle that we're going to couple on this TAM or fluorophore. The star is to remind me to say that this particular control experiment was with the unnatural tRNAs containing their unnatural anticodons, but the mRNA was fully natural. So you're getting GFP produced, that's the gene, and you're getting production because it's just a natural GFP. But there's no TAMR, there's no click, because there's no unnatural. Under the identical conditions, if your mRNA now has a natural codon, you see you get a gel shift. And then you get a click signal from TAMR. There's now unnatural content, the unnatural amino acid present. Maybe there's a little unnatural, maybe there's a little read-through still there from that band. But if you simply go to increasing concentrations of acetylysine, you can chase it away. It disappears with increasing concentration. What that means is it's not a problem with the decoding event, it's a problem with the charging of the tRNA by the synthetase. That's the step that's sensitive to the unnatural amino acid concentration. So that's Pete's problem, not ours. We wanted to choose a second codon, so we looked at GXC, same experiment. Here there's maybe a little more read-through when you don't have enough acetylysine, you're not producing enough tRNA. But all you got to do is add more acetylysine and you chase it away. By mass spec, both of these proteins are greater than 99% pure. So we were pretty excited. Here they are. These are the fully functional semi-synthetic organisms storing the increased information and retrieving it in the way of amino acid with an unnatural protein. Of course, they're green because they're producing GFP. That has nothing to do with the unnatural. But every one of the proteins that make these cells green also contains an unnatural amino acid. So there I'd like to end. So shown in red are the people in my group who worked on this project. I collaborated with Syntherex, this is a small biotech company that's focused on commercializing this technology. These agencies for funding, I think I mentioned the collaborators. And please, I'm in a competition with a friend of mine. Please follow me at Romesberg Lab. And if you follow Phil Barron, please unfollow him. So thank you.
Thank you, Professor Romesberg, for the very interesting talk. And I'm sure we're all going to follow him now. Okay, so we have time for a few questions. Panos.
So I have 2 questions, actually. So the first question is, doesn't E. coli try to get rid of this base pair?
So first of all, the fidelities I showed you, these are not error flips it out and can't cut it. Like one's a C nucleoside. There's no way it's cutting that. Remarkably, we published this earlier this year. MMR increases fidelity. When we knock out the MMR system, fidelity drops. And the reason we think that happens is what I just described. When you get a strand that's synthesized, if the unnatural base pair is in the synthesized strand, MMR can't cut it out. If a natural is in it, and it's probably forming the same screwed up structure that the correct pair forms. And so both are being recognized, but the system can cut out the natural and can't cut out the unnatural. So the fidelity actually goes, MMR actually has to increase the fidelity. But other than that, I mean, that data I showed you are what they are.
And so the second question is, when it's time to translate this unnatural base pair, do you have structural studies inside the ribosome? Because it's quite packed, the decoding set.
I didn't have time to discuss this. In fact, I didn't really have time to discuss any of the translation. What we've now done is we've looked at a lot of different codons. This is not the last pair we'll develop. We're still bringing them along. The translation data is just more SAR. And we're finding that a bunch of our codons translate really well. Some don't. First position, we can't modify.
Yes.
Third position's okay. Second position's the magic. So the two I showed you were both second position. And it's the one where you get the highest fidelity, most efficient decoding. Eukaryotic ribosomes are different. So we've now started to make a lot of success in eukaryotes and X cells. We actually have the system working. And if you look at the translation of different codons there are some similarities, but they're very different. There are great differences. The other thing that I didn't mention was all the codons that work well in E. coli are second position with NAM in the message. That's the one that works best. That's generally true for the eukaryotic ribosomes as well, except it tolerates TPT3 in several sequence contexts very well. So there are interesting commonalities and differences. And this is just SAR. That number one is going to guide further optimization. And two, it's just, I think, kind of a cool probe of the ribosome.
Thank you, Professor. In case you all have any